Welcome to the Biological Life Sciences channel. In today's video, we are going to see semi-conservative replication of DNA. This experiment was performed by Matthew Messelson and Franklin Stahl, in which they proved that DNA replicated semi-conservatively. So let's begin. There are basically three models for DNA replication. One is semi-conservative replication, next is conservative replication, and third is dispersive replication. In semi-conservative replication, two strands of DNA unwinds from each other, means the parent will have two strands and they unwinds, and each acts as a template for the synthesis of new strand. So the original copy of the parent is conserved, one copy. Complementary strand, this results in two DNA molecule with one original strand and one new strand. So this is semi-conservative because the one parent strand is conserved here. The next is conservative replication. In this model, DNA replication results in one molecule that consists of original both strands identical to the original molecule, and another molecule is formed using this molecule that consists of two new strands, exactly the same sequence as the original molecule. So this is called conservative replication. And the third one is dispersive replication. In this dispersive model, DNA replication results in two DNA molecules that are mixture or hybrid of parental and daughter DNA. In this model, each individual strand is a patchwork of original and new DNA. So the replication is dispersive here. So now let's see how these three strands are different. You can see the original strand here. In case of conservative one, the original strand will retain as it is, that is two strands of parents are retained, and a whole new copy of a new strand is synthesized. Both the strands here are new. So this is conservative one. The next one is semi-conservative, means the two strands of the parents are equally distributed among the offsprings, and using that single strand, the new template strand will be synthesized on both parts. So this is semi-conservative one. And the third one is dispersive one. Here patches of new DNA is used and the new strand is synthesized, and therefore this is called dispersive one, and it is not an proper DNA replication. Before going to the experiment, let's see the caesium chloride density gradient centrifugation. This is a particular centrifugation in which separate molecules such as DNA into bands. DNA is separated into bands by spinning them at high speed in the presence of another molecule such as caesium chloride. So this caesium chloride salt is used, its solution is used, and DNA to the level of different weight of the atoms can be separated here. So it forms a density gradient from bottom to the top to the bottom of the spinning tube or centrifuge. Density gradient centrifugation allows very small differences like those between heavy isotopes of nitrogen and light isotopes of nitrogen. Means in this kind of centrifugation, it can separate two strands even of the DNA which is having a normal nitrogen, that is N14, and a heavy isotope of nitrogen, that is N15. So there is a difference in a very few difference in both these DNA molecules. So even that can be separated in this kind of centrifugation. Messelson and Stahl knew that how often E. coli cell divided, means the division time of E. coli is 20 minutes. So in a very short time, we can get a number of samples of different generations of E. coli. So therefore they used E. coli here for this experiment. So they were able to collect these small samples at each generation and extract and purify the DNA. Then they measured the density of the DNA using density gradient centrifugation. Let's see the experiment. First of all, what they did, they grew the nitrogen, the E. coli on the heavy nitrogen containing medium. So what will happen, the nitrogen present in the medium will be taken up by the bacteria for its metabolic work. So in that process, it will utilize this heavy isotope of nitrogen, and which will form an integral part of the DNA of that bacteria. So what will happen, this DNA will be present everywhere in the bacteria that will be having N15 isotope of nitrogen. So now when the DNA is extracted and centrifuged to the equilibrium in caesium chloride density gradient, it will form a heavy nitrogen band here like this. So this band is from the original parent molecule that is grown on DNA, that is N15 containing medium. Okay, now the next generations are then grown on a different medium, that is a normal medium, nitrogen 14 containing medium. So this is normal nitrogen containing medium. So what should happen, some of the cells which are dividing at this stage will take up the normal nitrogen here. Okay, so it will form a hybrid strand. You can see the first generation, means after 20 minutes the first generation of E. coli formed by this organism which was used here, it showed like this. It showed two different bands. One is one band from the original parent, and new band is the newly synthesized one. Similarly, in the second offspring also it was similar. So after that they again grew this into the next tube, and next generation was grown, and in next generation it is seen that a totally new strand is synthesized of the other two offsprings which is formed from in the second generation daughter molecule. So here when the DNA is extracted, what will happen? It will form a different light band at the top. So when these three things are combined, you can see that the DNA which is formed in a hybrid forms a band at the center. You can see this band is at the center, this is the band at the top, and this is the band at the bottom. So what will happen? This will form a bond at the center, which shows that the DNA is replicating semi-conservatively. So let's see the conclusions of the experiment. Cells were grown for many generations in the medium only containing heavy nitrogen, that is N15. So the nitrogen in the air DNA was 15N, as shown by the single strand, that is blue, when centrifuged in caesium chloride density gradient. Now, once the cell has been transferred to the medium containing normal nitrogen, that is N14, cellular DNA isolated after one generation equilibrated at a higher position in the density band, as we saw in the last picture. Continuation of replication for second generation yielded two hybrid strands of DNA and two light strands of DNA, confirming the semi-conservative replication of DNA. So this shows that in offsprings, a parental strand is retained and a new strand is formed, hence the DNA is replicating semi-conservatively, and this experiment proved this. Thank you. Please press like button, share, comment and subscribe if you like the video. Thank you so much for watching.